My God. One time I saw a video of something eating little rat babies, but like it didn't click for a little bit, like because they're like little. No, like something. Like I don't remember. It was like something with no. It was like a raccoon or something with fingers. But it didn't click at all for like most of the video that it was eating rat babies and that's just something that I think about sometimes.
Okay. I know I said 5 minutes, so some people are probably wouldn't start about something.
I would.
So real quick,
I would find money.
so today is the last lab, like the last pile of information you have to catch and cram somewhere in your head. On Tuesday, we're going to do probably a longish lecture, and then Tuesday will be the last lecture portion for the lecture exam. So we're kind of wrapping it up here. Tuesday, during lab time, will be a chance for review. If you want to pull the sharks out and quiz yourself, if you want to pull a frog out, it's already dissected, quiz yourself, etc. Like I said, I'm going to use your sharks and frogs on the exam. So if you had a good shark or frog, it'll show up on the exam. So Tuesday, I'll do a little bit of a lab review. We'll kind of agree on what we're potentially going to cover. As usual, I drop massive hints as to what to expect, right? They probably want to listen to that. And then our lab exam is a week from today. We'll do the customary start at 9 o'clock, right? So we can start 1 hour later, so I can make sure that I have everything set up and that everyone gets here in time. And that'll take, because we have about 25 people left here, that'll take about an hour to do, and you'll have as much time as you need to go around and look things again. So that's it for lab. And then the lecture exam, we'll finish up our lecture content on osmoregulation and the kidney function on Tuesday. And then the following Tuesday, I said we'd get back our lab exams, review that, and then we'll have time for a lecture exam review, which is pretty short because we don't have much topics to cover. And that's it. That's all she wrote, right? So there you go. All right, let's go ahead then and get into the mammals here. And I don't have a whole bunch of slides. I just wanted to have a reference slide here. But one of the key features of mammals that we're going to look at here, the mammalia, is they have mammary glands, which are one of a number of subcutaneous glands they have. So one of the key features of mammals is they have subcutaneous, meaning they have glands under the skin. Most of the animals that we've seen so far, if they do have exocrine glands, will have it actually embedded in the skin. So in reptiles or in amphibians, they basically have this, some of you peeled the skin off the frog, and that's where any of those glands would be. Mammals have the glands under the skin, between the skin and the connective tissue that connects to the muscle. So in the rat today, they're not very mature. They do have mammary glands, but they're not really super developed because none of them are pregnant. But you will see salivary glands, another example. You know, when your salivary glands are active, sometimes you can kind of feel them here under your jaw. And so your salivary glands are located here. Your lacrimal glands, also your tear glands, are actually subcutaneous, transferring your tear ducts to your eyes. But anyway, subcutaneous glands is one feature. That's where the mammalia comes from, is mammary glands to nurse developing babies. Hair. Hair is actually a dermal feature. It's a eruption of a hair follicle from the skin, from the dermis. And so it's unique to mammals. We saw feathers in birds were a modified scale. That was a totally different type of feature. A scale feature is basically an external skin feature. Hair is a unique mammal feature. And then the last feature that's kind of interesting is endothermy. We're not really going to get a chance to talk about that too much or at all. But what's interesting is in mammals, mammals are able to regulate a constant body temperature even if the outside temperature is wildly fluctuating. So if it's cold out like it is today, or it's snowing out, even super cold, you're able to do metabolic changes that increase your metabolism that will generate more waste heat. And it's kind of interesting, this will all stem from your thyroid. The thyroid gland, which is just above your heart, and we'll see this in the rat, the thyroid gland, right on top of the heart. Basically, it's kind of like the gas pedal of your metabolism. And your thyroid, producing thyroxine, essentially tells the mitochondria in your cells to be less efficient. So rather than converting 34% of your cell food into cell energy, it'll convert more like 20 or 25 or even 15, and you generate more body heat as a waste of metabolism. So mammals are able to do this, and so are birds. Birds actually can do the same thing, but it's probably independent because birds can't really control it. Birds are either gas pedal fully on or gas pedal fully off. They can't fine tune their metabolism. But birds are also endothermic, meaning that they are able to regulate the body temperature when it's cold out. I think the presumption there is that enabled mammals to be active year-round and not go into hibernation or not be inactive when it's cold out. Also to colonize cold environments, etc. It's kind of a unique feature of mammals to have that endothermy there. And that's pretty much the main features that make animals unique. There are some mammals that have kind of reptile-like features. And so we do have some kind of, I'm not going to use the term missing link, but transitional species. And that would namely be these, what are called monotremes, these egg-laying mammals that occur in like Australia and New Zealand. Australia and New Zealand, Australia, I believe, is where the platypus occurs. The duck-billed platypus is an egg-laying mammal. And then there are a couple species of spiny echidna. I think one of them occurs in New Zealand, or maybe both in New Zealand. But anyway, these are egg-laying mammals that are taking basically the reptile plan for how they reproduce. They essentially put their embryo into an egg that's surrounded by an amniotic sac and a chorion and yolk and all that stuff. But what I want to point out is that the placental connection that other mammals have, the non-egg-laying mammals have, most mammals, basically it's an egg that's essentially retained internally. All the features are the same: the amniotic sac, the yolk, and the allantois, which is kind of the waste dump ground. But what's been replaced basically is a placental connection that connects the mother's circulatory system to the baby's circulatory system, and so we can have nutrient and gas exchange occurring there. But what's also interesting, just to reflect back, is these egg-laying mammals have nucleated blood cells. So remember I was saying with frog red blood cells have nuclei. If you look up on Google and look up duck-billed platypus blood, you can find pictures of it, and it looks like reptile blood or bird blood. It's really fascinating. So it is kind of interesting that there are a few remnant, what might have been kind of an example of a primitive mammal back in the Mesozoic. But mammals basically evolved sometime in the probably Jurassic or maybe very, very late Triassic is when mammals first appeared in the fossil record. But then obviously in the Cenozoic, the mammals radiated, and our current time, the Cenozoic, is definitely mammal time. So anyway, let's go ahead and take a look at our rat. I don't have any, one thing I want to point out though, I forgot to mention, is that the mammal skull is a unique design. I will bring this out here. The mammal skull, I'll bring our token mammal out here. This is what's called a synapse to the skull. So I left all the stuff out from last time so we can compare these. But the mammal skull has one temporal opening. And so it's called a synapse design. Let me go ahead and draw that here. The mammal skull, something like this. So here is our jaw, whatever. And there basically is a single jaw muscle that comes off the top of the head. there is a single temporal opening that basically the muscle goes through. So this is a one temporal opening or a channel, and this is called a synapse skull. So we mentioned that the turtles had an anapsid skull, where they basically have no temporal opening. The jaw muscles are basically internal. I mentioned that the reptiles and birds have a diapsid skull, where they have two pairs of jaw muscles that go through two temporal openings on each side. The mammal has a synapsid design that is basically one temporal opening for one jaw muscle. So this is a cheekbone, this is a zygomatic arch. But you can feel that on your body because your cheekbones basically here is where your jaw muscle originates right around the top of your temples. And if you open and close your mouth, when you put your hands on your temples, you can feel a jaw muscle. It basically goes under your cheekbones and connects to your jaw directly there. So that's that one temporal opening design is unique to mammals. And in the Paleozoic, there were reptiles that had this feature, so it's thought that mammals derive from a synapsid type reptile. But we won't talk too much about that. And so here's our token mammal. I just had it handy here, a little chipmunk, and or is this a ground squirrel? One of the two, chipmunk or ground squirrel. Oh, chipmunk. So anyway, you can take a look at that. And so anyway, on the test, I might have a mammal skull, probably this one, because this is probably the best one I have in the cabinet, and maybe our little chipmunk here, and ask you some questions that are unique mammal features. So for the rat dissection, what we're going to do is we'll cut the rat open the same way. The rat skin is very, very loose because these are fresh specimens. Also note that the skin of a mammal, in fact, one interesting tidbit of trivia is the largest organ of your body is your integument. Your skin is actually the largest technical organ of your body. And so it's very thick. And so what you'll notice is when you cut the rat open, you're probably going to end up cutting the skin open first. In years past, I had actually had students skin the rat. Now, if you want to do this, or if you know how to skin animals, it's actually pretty easy. But it's interesting that you can actually pull the entire skin off the rat. I had a student years ago who was a taxidermy person. He was really good at taxidermy, and he skinned the rat in like 20 seconds. Like, rat was down, and someone started a timer, and he had that rat skinned in 20 seconds. I was pretty blown away. It was a perfect skin. So if you want to try to skin the rat, this would be kind of an interesting feature to try. What you do basically is make an incision, cut from basically the genital opening to about where the throat is, and then essentially you peel the skin like taking a jacket off, and then take it off the hind legs and the tail first, and then pull it over its head like a cape. If you want to try that, I can help you out. If you don't want to skin the rat, you don't have to. But one year, I'll see if I can find it, we had a student bring in his sister's Barbie doll, and they put the skin on the Barbie. Kind of like a warrior Barbie fur coat kind of thing, you know? I'll see if I can find that somewhere. It's pretty twisted, so I don't like to let it get out there. I might have Tia knock on my door or something. But anyway, but what's interesting is the skin can be removed quite easily. So that integument is a key feature of mammal features. Very, very thick skin that has glands, extremities, etc., and hair. Okay, so cut the rat open. You're going to have to go through the rib cage, and I'll show you some features to try to avoid in the rib cage here. So let's see here. Let's go ahead and take a look at the rat in belly side up here. Here's our little rat. Okay. And
what is he drawing?
Again, we're going to be looking at, that's one cool thing about the vertebrates being at the end of the semester, is really vertebrates all have the same parts. It's like you're going to see the same features and sort of in the same location as in the shark. And that's what I really want to relay, is that vertebrates are really kind of all based on the same body plan, which is pretty cool. And so this lab exam probably you'll find ends up being kind of a bit of an easier exam, because we're just kind of seeing the same parts again and again, versus like comparing a horseshoe crab with a scorpion here. Okay, so you're going to go ahead and make an incision here. The rib cage here, you'll want to cut straight up the middle of the rib cage, because there is a subclavian artery that occurs off the heart, right, going to the arms. You have a couple major arteries in your body that if you get damaged or injured, you can bleed out very quickly. One of them is your subclavian. Like if you get a stab wound or a gunshot wound here under your armpit, you can quickly bleed to death because you have basically high blood pressure and it's a direct line from your heart. Same for your femoral artery. Arteries in your groin going to your legs. There's a major artery here as well, but you probably won't cut that one. But do be careful around the arm, because there is kind of a chance of bleed out. But what's interesting is because the rats are dead and the heart's no longer pumping, there's not much blood pressure, so you can actually do this section with very, very little blood coming out, as long as you're not breaking any blood vessels. So anyway, okay, so let's go ahead, and I want to point out a key feature here that I want you to see, probably will certainly be a test question. And that is the big dividing point in the rat will be separation between the peritoneal or abdominal cavity and the pulmonary or thoracic cavity, and that is the diaphragm. So, because the diaphragm is let me get that larger so you can see the spelling. Because the diaphragm is a unique feature of mammals, I'll probably certainly have that one pinned. And it has weird spelling. The diaphragm is a skeletal muscle that separates the lung and heart cavity from the abdominal cavity. And when it's relaxed, there's like a little tendon here that kind of pulls it up like a dome. And when it contracts, it flattens out, increasing the volume of the lung cavity, inflating the lungs. And one of the things I'll have you do at the very end of the lab is I'm going to have you inflate the lungs with a pipette, and then you can see how the lungs inflate, ventilate the lungs. But this is a really interesting, unique way to get lungs ventilated, very unique to mammals. So certainly one of the questions on the rat, we'll have one rat on the test, and diaphragm will be one of the questions, certainly one of the answers there. I might even label it letter D for you, just to help you out, right? Christmas comes early. Okay, so anyway, let's go ahead and take a look at the digestive tract, because through the diaphragm comes the stomach. So the stomach is in the same spot as it is in the shark and the frog, basically the same kind of left side location. Your stomach's basically kind of right around here. And then the stomach has a pyloric valve, which regulates the passage of food from the stomach into the duodenum, if you're British, or duodenum if you're American. So here's our duodenum. Okay? That little S-bend, same exact S-bend that we saw in the frog and the shark. There is a new gut feature that's in mammals that's not in any other vertebrate, but I'm not going to pin it because it's really hard to find in a rat. But there is a segment of the intestine called the jejunum. And how you can, so I'm not going to ask on the test because it's hard to really see it on the test in 2 minutes. But the way to find the jejunum is, the next part of the intestine is the ileum. And the ileum is the absorptive, what we call the small intestine. And what you'll see is the ileum has lots of vascular tissue associated with it. This is the ileum, the absorptive intestine. So you'll see lots of blood vessels going to the ileum. the point at which it looks like it's no longer duodenum, but there's no blood vessels, that's where the jejunum is. And the name jejunum, I'll just go ahead and write this down. This is all stuff that's not on the test, but I think it's interesting, which students hate, but I know this is the fun stuff. What jejunum means in Latin is fasting. So juju in Latin means to fast or to starve and not to eat. The jejunum is a section of the gut tract that never has any food in it. So the stomach has food in it, the duodenum usually has food in it. What happens in the duodenum is the pancreas secretes bicarbonate onto the food, neutralizing stomach acids. So you have stomach acids in your stomach that can range from pH 1 to 3 or so, very acidic. And in the duodenum, the pancreas will convert that food into more of a neutral, maybe a little bit basic, like 7.1, 7.2 pH food material. Anyway, this jejunum is kind of like a transition point where the food is transferred from the duodenum to the ileum. So it never has any food in it. So if you want to find it in the rat, it's probably about maybe a centimeter or two long, maybe a centimeter and a half long, very short. You can kind of see it's a part of the intestine that's very thin, and you can almost see through it if there was light behind it. There's no food there. But it's really hard for me to get a pin in that, and for that to be consistent throughout a whole test. So I'm not going to put the jejunum on the test. I'm just going to put it there so you know there's a new feature that mammals have that the amphibian and the fish did. Another feature that we're going to look at that I probably will ask is there is a very large cecum. The cecum is a pocket off of the junction between the ileum and the colon. And the colon, which is often called the large intestine, is pretty easy to recognize because you are probably going to see fecal pellets developing in the colon. So you'll see basically firm fecal pellets as it approaches the anus, but the pellets will probably be a little bit more mushy as they approach the cecum. So you'll see in the colon here that the food pellets basically get more and more firm or solid as they get to the end. What's happening is the colon is much more elaborate, much more long colon than was in the frog, because these rats are retaining a lot more water. The rat's able to live on land full time, doesn't have to go to the water. They do have to drink water, but they are able to tolerate much drier environments than an amphibian because they have this large intestine to be able to resorb water from the food stuff. And you can see those fecal pellets kind of get more and more firm as they get closer to the colon. Sound good? Okay, so I do like to have the cecum on the test because it's going to be a new thing. One thing I will point out is probably avoid poking the cecum or breaking it because it smells like the worst fart you've ever smelled in your life, right? Or you can blame your neighbor. But anyway, the cecum is an area of microbial storage. It's basically where a lot of microbes are being stored to help the rat digest a high fiber diet. So there's lots of bacteria in the cecum. And when it's broken open, it smells really horrible. Similar with the stomach. The stomach also smells pretty bad when it's open up. So just kind of avoid that. Or it'll clear your table pretty good. Okay, any questions on the gut tract? This is basically, pretty easy. Most of it's very similar to the shark and the frog, but we do have two unique new things. This one won't know that, this one probably will. Okay, then we have our organs that are associated with this. We have the pancreas in the usual pancreas location. Pancreas. Again, the pancreas has all kinds of functions. One is to secrete bicarbonates so that the food gets neutralized from stomach acids. The other is it regulates blood sugar. It has proteases. Pancreas is a very important organ. We also have a spleen that's located here. The spleen in the rat is kind of strap-like. So it'll look like the same color as liver, but it'll be like more of a strap-like organ. Whereas the frog, it looks kind of like cranberry sized thing, and the shark, it looks kind of like a little triangle or like a wedge type structure. The liver is, the largest internal organ here, and I'm not going to really draw it to cover things up. Interestingly enough, the rats don't have a gallbladder, and I don't know why. The gallbladder's function is to secrete bile, store bile for secreting onto the duodenum, and bile is a fat breakup emulsifier, a salt that breaks fat down in small droplets. It might be because rodents are mostly herbivores eating a high cellulose diet that maybe they lost their gallbladders, but other herbivores like sheep and horses have gallbladders, so why rats don't have gallbladders, I do not know. But that's an interesting note. So of course I wouldn't ask gallbladder on a rat because they don't have one, right? So the liver is organ there. I probably won't ask the liver on the rat because I have other things I can ask. Okay, and then finally, the kidneys are located here in the back body wall, and these are really well-developed kidneys. Anyway, before you finish your dissection, I would like you to section one of these kidneys. So don't cut it out right away. Like don't rip the rat open and then suddenly cut a kidney out. What will happen is there is a major artery and vein that supplies blood to and collects blood from the kidneys. It's basically right off dorsal aorta and dorsal cardinal vein. The kidneys basically supply blood to those two major vessels. So when you cut the kidney out, lots of blood will fill up this cavity. So wait till the very end after you've seen everything else, and then you can cut the kidney out. But basically the kidney, we're going to cover this on Tuesday. It'd be kind of cool if you saw what the parts are. And the parts basically are, we have the renal artery and vein. So this is the renal artery. I mean, you won't really be able to tell this, but that's basically the blood supply to the kidney. So to cut the kidney open, you're going to do what's like a transverse section. You're going to basically, and they look like kidney beans. That's where of course the kidney bean got its name. It looks a lot like a kidney. You're going to cut it kind of like this, so you can get a section. So your view will be like that. Anyway, the kidney has a couple main parts that we're going to talk about on Tuesday. So there is an outer cortex, which is where the filtration happens, the glomerular filtration. So there's a cortex, and then there is an inner medulla region. And you can see these in the section. So if you cut the kidney open like this, you'll see kind of what looks like porous material or kind of a lighter area on the outside of the kidney. And you'll see maybe a darker striated or streaked area on the inside of the kidney. So note those. I'm not going to ask these on the lab test, but on the lecture exam, there will be a question on the kidney anatomy because we're going to talk about that Tuesday and how the kidney functions. So you will have to know what the cortex and medulla is in 2 weeks. So I think it's kind of cool to check it out in the rat since we have it handy. Okay, I think that, one last organ that you probably are going to see, but I won't ask it on the test, is the bladder. There will be a urinary bladder located, let me get a good bladder cover. This is a bladder that is located right in front of or ventral to the rectal opening. And if you're really good, you can actually see the ureters, which are the urine supply that comes from the kidneys to the bladder. I won't ask them because they're so thin I can't put a pin in them, but in the back body wall connecting the kidney to the bladder, you'll actually see these little ureters. And then you'll wonder how in the world, if you have ureters that are probably, the diameter of a millimeter or two in diameter, you'll wonder how in the world can you pass kidney stones through those. That's why kidney stones are so painful if you have them, is those ureters are very, very small and they're very thin and pretty easily damaged. But I won't label them, but just go ahead and see if you can see those. Kidney for sure, though, would be a good fair question on the exam. Okay? That's all the abdominal cavity features. Let's go ahead now and look at the pulmonary and the cardiac features. So the heart is a 4-chamber heart. You can actually see in the ventricle the two sides. There's actually kind of like a seam or like a suture that goes up the middle. And what you'll see is the left ventricle is a little bigger than the right ventricle because the left ventricle is supplying blood to the body. The right ventricle is supplying blood to the lungs. So the left ventricle is a little bit bigger. So you'll see the little groove a little off to the side so that the right side, the rat right, is a little smaller and the left side, the rat left, is a little bigger. You're probably not going to see the aortic arch, but if you do, that'd be awesome. But basically, the blood system does something like this. There is an aortic arch that supplies blood to the head via the carotid arteries. There's also a subclavian that comes off that goes to the forelimbs. But basically, the blood supply to the head and the forelimbs comes off of this arch. And then the aortic arch goes through the diaphragm, and then the dorsal aorta basically is in the back of the body. I'm not going to draw it there because it kind of clutters it up. But dorsal aorta basically is the main blood supply that supplies blood to the kidneys, for example. So what's interesting is the kidneys function by blood pressure. It's your blood pressure that regulates filtration in your kidney, one mechanism of filtration. And so that's why if you lose blood or have low blood pressure, sometimes you have to go on kidney dialysis because your kidneys require a high pressure to keep that filtration happening. But anyway, so there's the ventricles. And then the atria, you can see there will be a right atrium and a left atrium. And those are pretty obvious. And then right behind the atria will be the lungs. And they're deflated now because the rat's no longer doing respiration. But what I'll have you do near the end of the session, once you've seen everything, you need to really cut up into the throat cavity and expose the trachea. So the trachea is like a tube with cartilage rings that support it. And you can feel your own trachea here. You can feel it's kind of ribbed. And so you'll have to kind of cut through the thyroid gland, which is here on top of the heart and the throat area, and expose the trachea. And basically what you do is a tracheotomy. I will get some clean scissors, some scissors that aren't funky over here that have been used to cut everything open. I'll go get some clean scissors from my office that I don't use for cutting critters open. And what you're going to do basically is cut the top of one of these pipettes off, just snip off the top of the pipette, and now you have like a straw. And you're going to do a little cut in the trachea, like a longitudinal incision, and you put the tube in there, and then make sure everyone's watching. One person can blow into the tube, and you can inflate the rat lungs. And this is pretty cool because what's amazing is just how big of a volume these lungs have the potential to be. And it's mind-blowing. So some people use their phones and video, like that's totally cool if you want to do that. But it's pretty remarkable to see the lungs inflate in that chest cavity, just to realize how much capacity they have for respiration. Okay? And with this being clean, nothing should come back at you. That's why we use the big tube, because I've never seen anything come back like blood or anything. So But we do want to have only one person blowing on the straw and not sharing that. We don't want spreading diseases or anything. Okay, on the dissection or on the lab exam, lungs could be a possible option. Or if I were to have a pin in the ventricle, I would want you to tell me whether it was the left ventricle or the right ventricle. Okay, so I'm not going to really label it here because everything's being kind of cluttered up right now. But you should realize the ventricle here has two halves, whereas the frog ventricle and the shark ventricle only has one. That's a big difference. Sound good? Okay, I will do this. I have the rats in a tin, like a chamber or whatever. I'm going to go ahead and clear off some space here. We'll go ahead and put the rats over here. I have 6. I made sure I got 6 this time. I feel so bad about that frog. We worked it out. But anyway, here is, we'll put these over here. The skull and the chipmunk. Anyway, so we're going to use our dissection pans here. Because these are fresh, we can't put them in the trash or whatever. So when you're all done, wrap your rat up in a paper towel, like a little rat burrito, and then put it back in the bin here. And what we do is we freeze them so that they don't go stinky, and then we get rid of them all at one time so they don't sit in the trash. Because otherwise, if we put them in the trash or any parts in the trash, the janitor who shows up late at night will be really mad at smelly trash. So we'll put the rat back in the bin here, and there you go. And then also make sure that you clean off your tabletops, and certainly help me out by rinsing out your pans very well, so the blood or whatever tissue that's left in the pans doesn't get putrid. All right, let me go ahead and grab the rats. I think they've had enough time for the big sleep. I use chloroform to euthanize them.
I remember when my parents used to call it that.